Everyone. Thank you so much for being with us today. We're incredibly fortunate to have Dr. James Adam with us today. Welcome, Dr. Adams.
Welcome.
Thank you. So I just want to give the audience a little bit of a background, even though we know a lot about you, but it's always good to do a little background, and then you have a wonderful presentation to share with us. So Dr. James Adams has a PhD in materials engineering, but for the last 20 years has focused his research on the biological causes of autism and how to treat it. He has published over 200 peer-reviewed scientific articles, including over 50 on autism. He has led many clinical trials, including nutritional supplementation, microbiota, transplant, and detoxification. And today, Dr. Adams is here to discuss his study on the MTT with autism and other GI distress. And we'd love to hear how everything's going.
Great, thanks very much. And if people want more information, they can go to our website, autism.asu.edu, and there's more information there about our studies and how to sign up for future studies. So I need to make some disclosures first. Several of my collaborators and I have pending or approved grants for the use of MTT or probiotics for a variety of conditions, including autism. Several of us have received research funding from Christovo, which is now Finch Therapeutics, for our FMT research. And several of us are also part-time consultants trying to help Finch with its research. And we receive grants from them. I need to point out that MTT is an investigational treatment and is not approved by the FDA for autism or for any condition, but that's why we're doing the research. Also, several of us are part owners of Autism Diagnostics, which is developing Metabolomics tests for diagnosing and treating autism, and we have several patents pending in this area, and I'll talk more about that later. So just a little bit of background. 20 years ago, I was very excited by a research study by Sandler and others, in which they showed that giving a special antibiotic, vancomycin, was very effective in reducing GI problems in children with autism, but also greatly improved behavioral symptoms. And this is a special antibiotic. It's a very potent one, but it's also very safe because it's not absorbed into the rest of the body. It just stays primarily in the gut. Unfortunately, after 8 weeks, they then stopped treatment, and then within a few weeks, all of the gains in gut symptoms and autism symptoms were lost within a few weeks. So this is just a little chart showing at baseline, by definition, they're all at 0. During treatment, there's a nice increase in autism symptoms. And then after treatment stopped, unfortunately, within just a few weeks, even though some of the children were taking standard probiotics, they lost most of their benefits of symptoms. So they did also notice that a number of the children, about half, had a temporary increase in hyperactivity right at the start of Vanco, usually lasting just a few days. We think that's because as the vancomycin is killing off harmful bacteria, those bacteria are releasing their toxins all at once. There has been a major survey by the Autism Research Institute showing that whereas antifungals seem about half the time to significantly improve symptoms, general antibiotics tend to worsen symptoms about 30% of the time, and only about 12% of the time do they help. So it seems there's something special about vancomycin and the bacteria it targets. We published a study 10 years ago in which we showed that children with autism who had GI problems had worse symptoms. So blue are the children with autism without much GI symptoms, and red are the children with autism who had significant GI symptoms. And what we see is that higher scores in every category. So the children with autism who had GI symptoms had worse speech, worse social problems, worse sensory and cognitive problems, and worse physical and behavioral problems. So our thinking then is that if we could treat those GI problems, we might be able to reduce their symptoms down to the group in blue. And so that was our simple thinking. We also did a study with my collaborator, Rosie Crouch-Malnick-Brown, and Daewoo Kang, and they've helped with a number of the projects I'm going to discuss today, in which we looked at the microbiome diversity. So your gut bacteria, most of them are healthy. You have typically of order 1,000 different species in your gut. It's generally believed that the higher diversity, the more types of species, the healthier your gut is. And we found, looking at both the total number of species, CHA1 or bacteria, we found that kids with autism in red had fewer species than the children who were typically developing. And it may not look like a big difference, but this is a log scale. So in general, they had about 25% lower diversity. In other words, they're missing several hundred bacteria compared to typical children. Also, when we look at the PD index, which is a measure of the diversity of the genes in the bacteria, we found that the bacterial in kids with autism had much lower diversity in their genes as well. So two different measures finding the same thing. Kids with autism seem to be missing several hundred species of bacteria. In particular, what was strikingly different was almost all of the children with autism were missing certain species of Prevotella. So this chart shows on the left-hand axis different species of Prevotella. And these first a few children with autism had, and many typical children had. But for these other species highlighted in green, we saw that none of the children with autism had those, whereas many of the typical children did have them. And it didn't matter if the children had GI symptoms or not. This group did not have significant GI symptoms. This group did. And we found that in either case, they're still missing the Prevotella. And what's special about Prevotella is that in studies of children in Africa on traditional, very healthy diets, and these children had minimal GI problems, these diets were rich in carbohydrates and fiber and non-animal protein, so mostly whole grains, legumes or beans, and vegetables. Half of their gut bacteria were Prevotella versus of order 1% or less in the children in Europe on traditional Western diets. And in the US, it's similar. So if we think about healthy children having a high level, half their bacteria Prevotella, typical children in Western diets having of order 1% or less are Prevotella, and children with autism about 10 times lower. And we found that Prevotella anti-correlated with many bad bacteria. So when Prevotella was high, many pathogens were low, and vice versa. So we don't know which is cause and effect, but basically it's a biomarker that these children with autism do not have healthy gut bacteria. And so to conclude, we found that children with autism have much less diverse microflora. They're missing several hundred of the typical 1,000 species in their gut, and lower diversity is associated with a less healthy gut. And Prevotella was especially lower in children with autism. And in future, we'd like to consider as a potential probiotic, but currently it's at least a health-specific biomarker. So there have been many studies of probiotics in children with autism. These are generally small, limited studies. Most are open label studies. I don't have time to go over them now, but I'm just letting you be aware that in general, they do suggest that there is some benefit to the use of probiotics, but more research is needed to really confirm that and to figure out which probiotics are needed. So in terms of the treatment implications, if children with autism are missing several hundred species, and one child with autism is missing these several hundred, and this child with autism is missing a different set of several hundred, then we probably want a very broad spectrum probiotic. The problem with most commercial probiotics is they only contain a few species of typically 1 to 10 different species of bacteria. And generally, these are bacteria that have been raised in milk cultures. So they do great growing in milk, which is exposed to air. But in your gut, we have primarily anaerobic bacteria, bacteria that are killed by the presence of oxygen. So we have very different bacteria in our gut. So when you take probiotics, they can temporarily help in some cases, but they will generally, when you stop taking them, they'll disappear in 1 to 2 weeks from your gut. They don't generally engraft there. An alternative approach is using fecal transplant, taking bacteria from very healthy donors and giving those to children with autism. So this might sound like a very radical treatment, but in fact, it's become a very powerful and widely accepted treatment for a very nasty infection called Clostridium difficile. C. diff infections cause life-threatening diarrhea in about 500,000 Americans each year, and it kills about 29,000 Americans each year. Antibiotics can treat it, but it often comes back. And when it comes back, the person just becomes weaker and weaker until they may die if they can't fight off this terrible infection. What's amazing is that fecal transplant, just taking raw poop from a healthy person and giving it to a sick person, will generally cure these C. diff infections in 2 to 3 days, and with just 1 to 2 doses, and about 90% success. It's just miraculous how effective it is. And even better, once you've been treated, it's almost unheard of for these C. diff infections to return. You seem to be cured for life. So therefore, it's not surprising that FMT is now used by hundreds of hospitals across the US to treat over 50,000 people with C. diff. But it's not yet approved by the FDA. It's in a very special regulatory environment, so phase 2 trials are being conducted. So the FDA is allowing it to be used for patients with C. diff that has recurred after antibiotics were tried, but it can't be used for any other condition currently because of FDA regulations. So in Australia, one of our collaborators, Dr. Thomas Brody, who's a gastroenterologist, he's been one of the pioneers on FMT. He's written a number of papers on it. His group has now treated over 15,000 people, including 9 children with autism. And I'd like to share with you comments he had. Basically, unlike with most of his patients where just 1 or 2 doses is sufficient, he found children with autism are much harder to treat than other groups. And therefore, instead of just 1 or 2 treatments, he treated them every day for 3 months. And what he reported, and this is a direct quote from him with his permission, after 3 months of the cultured microbiota, there was a substantial improvement in bowel function, and the parents noticed a substantial reduction in the odor of their stool. Several children started the study with abdominal cramps, which disrupted their sleep. By the end of the treatment, their abdominal cramps were reduced. They were able to sleep throughout the night. Parents reported marked increase in vocabulary, clear improvement in task performance, new ability to listen to parents' requests. So as a gastroenterologist, he was very surprised to hear that treating the gut could improve these autism-related symptoms. And so we worked with him and used his clinical experience to design a treatment protocol. And we submitted that plan to the FDA, and the FDA approved us to go ahead and do a study of it for children with autism. We give it a special name, microbiota transplant, because it involves several steps. And so the first part of our treatment protocol is 2 weeks of oral vancomycin, that special antibiotic that seemed very helpful to children with autism temporarily. The idea is that will kill off harmful bacteria. And then we do a day of fasting and then a very complete bowel cleanse, just like you'd have for a colonoscopy, to try to remove as much remaining bacteria as possible. And then do high-dose microbiota, followed then by 7 to 8 weeks low-dose microbiota. The microbiota we used are very highly purified. So we take human stool and it's extensively purified by our collaborators at University of Minnesota until it's just 99% bacteria. And we screen our donors very, very carefully so that only the 10% healthiest people in the US are allowed to be donors. And so the bacteria that we extract is basically like a super probiotic with 1,000 plus species of bacteria. And we believe that's very, very powerful. So in our study, we enrolled 18 children, ages 7 to 16 years. We wanted to enroll younger children, but the FDA said, no, let's try the older children first. These children all had moderate to severe GI problems. Most of them had constipation, sometimes alternating with diarrhea, and some had only diarrhea. We excluded very low functioning children who were not able to assent. They had to, because this was our first investigational study, they had to be able to agree to participate. And all of these children, we were surprised to find, had had these GI problems since infancy. The parents reported they had never had a normal GI health since infancy. We looked at what were factors as to why these children developed these GI problems, and we think we discovered a number of important clues. You inherit most of your microbiome from your mother as part of the birth process. And it turns out that the mothers of kids with autism, compared to the mothers of typical kids, reported lower fiber intake. And fiber is very important because your body converts it to butyrate, which is the main food for the gut. So eating enough fiber is very important. And we found that children with autism also tended to have somewhat lower fiber consumption. The children with autism in our group tended to be born by C-section. And when you're born by C-section, then you inherit a somewhat different type of bacteria. Normally through a normal vaginal birth process, you inherit a lot more of mother's bacteria. Also, we found the children with autism breastfed for a much shorter period, 3 months compared to about 9 months for the typically developing children. And breastfeeding is very important because mother's milk has a special ingredient in it, which promotes a certain special type of bacteria called B. infantis, named after the infant. And that's because only this special bacteria, B. infantis, can consume this special ingredient in mother's milk. And so that helps promote a very healthy gut bacteria. And finally, we found that children with autism had higher usage of oral antibiotics, which again is a risk factor. Every time you take a round of antibiotics, they disrupt your gut bacteria somewhat. Often it recovers, but maybe not fully. So we think these reasons largely explained why these children with autism had these GI symptoms to begin with. In terms of compliance in our study, every one of the 18 children who started the treatment completed it, which is very rare. We're very pleased we didn't have any dropouts. The hardest part of the study was the bowel cleanse. It didn't taste very good. And so a few children didn't drink quite all of it. And in a few cases, the fasting was not complete. A few of the children snuck a cookie here or there during the fasting period. The oral high-dose microbiota therapy was very well tolerated in 12 of 13 children, but one child had some nausea, so they were switched to the rectal dose. And the rectal dose was done as a deep enema over about an hour. And those children all tolerated that very well and were able to maintain the bacteria in their gut for several hours or more. And then when we switched to the oral low dose, that was very well tolerated. During the low dose period, they also received Prilosec, a stomach acid suppressant. And that's important because otherwise the stomach acid in your gut would normally destroy a lot of the bacteria that you're consuming. So your stomach acid is a great defense against toxic bacteria, but it also destroys most of the probiotics that you take. In terms of safety, we did have one child who developed a rash from the vancomycin, which had a natural orange flavoring. But when we replaced it with an unflavored version, then the rash disappeared. No significant adverse effects from the microbiota. No long-term concerns from our blood safety testing. Just some temporary, very minor increases in liver enzymes after the vancomycin. We returned to normal within a couple weeks. The vancomycin itself, similar to the previous study, 12 of the 18 children had a worsening of behavior for a few days. usually started the first 1 to 4 days of the Vanco, lasted only 1 to 3 days in most cases. One participant did have symptoms lasting for about 3 weeks. In 7 cases, we saw mild to moderate hyperactivity. In 5 cases, we saw mild to moderate increase in tantruming or aggression. But then those symptoms disappeared, usually in just a few days. And then GI symptoms and autism symptoms slowly began improving. It took about 5 weeks or more for GI symptoms and about 10 weeks for the autism symptoms. We found that this temporary adverse effect to the Vanco occurred primarily in the children who had worse of those symptoms to begin with. So presumably they had worse of the bad bacteria, so they're going to have worse of a die-off reaction. That's our assumption. The initial results of the study were very exciting. We saw initially an 80% reduction in GI symptoms, and then we waited 8 weeks to see if, after stopping treatment, to see if those benefits would remain, or with Vanco alone, if they'd be lost. And we found that those benefits remained 8 weeks after treatment stopped. They were very stable. Also, we saw about a 23% improvement in autism symptoms by the end of treatment. And again, that was stable 8 weeks later. So unlike with Vanco alone, these benefits with Vanco plus microbiota transplant, those benefits were stable. So we think that the microbiota really helped result in some long-term benefits. And also we saw the microbiome diversity improve from about 25% lower than normal into the normal range. We were very excited by this. We published our results. We thought we were done. This is very exciting results. And then 3 different parents came up to me and said, you know, my child's doing better than ever. This is about a year after we'd completed the study. So after the 3rd parent told us that, we were lucky enough to raise some additional funding from Finch and to do a follow-up study. And what we found, to our surprise, is that there was major improvements in autism symptoms by the families. In general, there have been no adverse side effects 2 years later, except one participant had some temporary worsening of behavior, lasted for about 6 months before improving. But we do have some limitations to this follow-up. The 3 participants did change their psychomed usage. 4 participants changed their nutritional supplements and or changed their diet. But our impression and the parents' impression is that most of the changes that observed post-MTT were due to the MTT itself. One exception is we did have one girl who had started puberty, and that led to a significant worsening of her behavior. In terms of long-term results, the GI improvements lasted for most, but not all participants. There was initially an 80% reduction, and 2 years later, it was still at 59%. We'd had one child who had had a strep infection right at the end of treatment, and they took antibiotics, and they lost all their GI benefits. So we know to be cautious with regular antibiotics afterwards. The autism symptoms generally continued to improve. Most of the parents reported just slow, steady improvement. Just seemed like their child was better able to learn new skills, new language, appropriate behavior, and just saw overall good improvements. According to our CARS expert, our expert autism evaluator, there's a 24% reduction at the end of treatment versus 47% reduction 2 years later. Again, this is an open label trial, so there's some placebo effect, but we think much of these effects are real. So if we look in a little bit more detail, this is the GSRS, the severity scale of GI symptoms. A 1 means absolutely no symptoms at all. So we see at the start of the study, many children had GI problems. We saw a big improvement by the end of treatment. We saw 8 weeks later, those benefits generally continued. And so by this point, 17 out of 18 participants had had improvement in GI symptoms. And then 2 years later, most, but not all, There are a few participants who lost some of their GI benefits, but most continued to have very good GI benefits. If we look at autism symptoms, we see that no one's symptoms worsened, and many of the children, most of them had very good improvements. So this is the baseline score in the CARS. Above a 35 means severe autism. This is the range from mild to moderate, and below here is below the cutoff for autism. 15 is an absolutely perfectly normal child, the lowest score you can get. And what we see is that after end of treatment, there'd been some improvement. 8 weeks later, it was stable. 2 years later, even more improvement. Another way to put it is that at baseline, over 80% of our participants were rated as severe. At 2-year follow-up, less than 20% were severe, 40% were mild to moderate, and about 45% were below the cutoff for autism. They still had some symptoms, but greatly improved. The parents also filled out their own questionnaire called the SRS, and they reported similar findings as well. At the start of the study, about 90% were severe. By the end of the study, about half were still severe, but about a third were mild to moderate, and about 20% below the cutoff for autism. So maybe not quite as much improvement on this parent-rated scale, but again, very, very promising. We find also that there's a very strong correlation between improvement in GI symptoms and improvement in autism symptoms. So the children who had, say, an 80% reduction in their GI symptoms, which was the average, also had a very good improvement in their autism symptoms. But there are a few children who did not have much improvement in autism symptoms or much improvement in GI symptoms. So just one didn't show much improvement in either GI or autism symptoms. In general, also, we looked at the microbiome diversity. And as I mentioned at the start of the study, the children with autism had lower levels compared to the dotted green line, which is for typical children. By the end of treatment, their microbiome diversity had increased to normal levels. Very surprising, at the end of 2 years, their microbiome diversity was above that of typical children. They actually had a higher microbiome diversity. So this suggests that this treatment is probably going to be a very long-lasting treatment if we're seeing this great improvement in microbiome diversity 2 years after treatment stopped. This is another slide showing engraftment. That means at the start of the study, the children were not very similar to their donor. After initial dose, they became more similar to their donor. Lower score means more similar. At the end of treatment, more similar. After 8 weeks after treatment, more similar. 2 years later, they're no longer very similar to their donor, but again, they still have that very high diversity. So their gut bacteria have presumably adapted to their own personal diet and body. We also found some good improvements in gut bacteria. Bifidobacteria, which is an important bacteria commonly used in probiotics. At the start of the study, the children with autism were low compared to the dotted green line, which is for typical kids. And after treatment, it improved quite a bit, and it stayed improved quite a bit 2 years later after treatment had stopped. Prevotella was undetectable in almost in all but 3 of the children with autism. And after treatment, it jumped way up and remained quite elevated even 2 years after treatment had stopped. So again, that's a good clue that we've restored a healthy microbiome in most of these children. We also saw a big increase in desulfo Vibrio, but there's some debate whether or not that's a benefit or not. So just to summarize, what we observed is that children with autism had abnormal gut bacteria, including a lack of a variety of beneficial bacteria, especially Prevotella. We found that microbiota transplant seems to be a very promising method based on this small initial study to restore normal gut bacteria and improve gut symptoms and improve autism symptoms with minimal adverse effects. And it seems these benefits are long-lasting. 2 years after treatment, it seems that most of these benefits are still holding. The autism benefits have just continued to improve. But this was an open-label study and a small study. So we need to do a randomized, double-blind, placebo-controlled study to verify these results. So based on our research, the FDA granted fast track status to MTT for autism. That means they recognize it's a potentially promising treatment for an unmet need, because right now there is no FDA-approved treatment for the core symptoms of autism. But we still need phase 2 and phase 3 trials for this to become approved by the FDA. So currently, we're doing an adult study with our collaborator, Richard Fry at Phoenix Children's Hospital. This is a randomized, double-blind, placebo-controlled trial. We've already treated 35 participants. We have another 10 who are being treated right now. I think it's about 15 actually. And we plan to enroll up to another 37 if we can raise the funding for that. We also are very lucky, due to many autism families raising funds for us, to completely fund a study for children with autism. It's a randomized, double-blind, placebo-controlled trial. We hope to enroll 50 children, and we've already started treating the first 2 groups of children. So again, this will give us more evidence between these 2 studies for this. But because of COVID, that has hurt our budgets quite a bit. So it's not clear we'll be able to reach the full number of participants in these 2 studies. And then we're also doing a study for children with Pitt-Hopkins. It's a rare single gene disorder that involves very severe autism, very severe GI problems. And so we're doing a randomized, double-blind, placebo-controlled study of that with funding from the Pitt-Hopkins families. And we've already started treating our first group of children there, and actually getting close to the end of treatment. So our goals of these studies are to demonstrate that this treatment hopefully is effective both for GI symptoms and for autism symptoms. Our first study guessed about dosing and duration. We're now going to a higher dosing for the maintenance dose and a longer duration to see if we can have even more benefit. So we have room to still improve on this therapy even more. Right now in our children and adult studies, half the children and adults are receiving vancomycin and half will not, because we know, although vanco is very effective at killing off harmful bacteria, it also kills off some of your beneficial bacteria. So we aren't sure if vancomycin helps or hurts, or it just doesn't matter. So we're trying to answer that question in these studies. And very importantly, we're trying to figure out which bacteria are changing and the effect they have on metabolism. So as I mentioned, my collaborators are Rosie Krajmalnik-Brown and her team are hard at work looking at this data and analyzing the results. So we have published a couple papers on beyond our initial work. One is for clinicians, some guidance and thoughts about where research needs to go in future. And what I want to talk with you about next is some very interesting results we did looking at the blood and metabolites in the blood of children with autism before and after microbiota transfer, because we want to try to figure out what exactly are the gut bacteria doing and what effect are they having on the rest of the body, and what effect does the treatment have on that. So we used a very advanced method called Metabolomics to measure 621 different metabolites or chemicals in the blood of children in our first study. And we found of these 621 metabolites, 11 were very different in the children with autism who had GI symptoms versus the typical children who did not have GI symptoms. So again, we don't know if the difference is due to the autism or the GI symptoms or both. Of these 11 metabolites, we found with just 3 to 5, we could differentiate the kids with autism versus the typical kids with nearly 100% accuracy. So this is just a chart showing that a 3D plot, we're plotting 3 of these metabolites, IMP, sarcosine, and tyraminosulfate. The red dots are the kids with autism. The blue X's are the typically developing children. And you can see there's no overlap between these two groups. Just with 3 metabolites alone, we have 100% ability to differentiate with just a drop of blood, which is a child with autism with GI symptoms and which is a typically developing child without autism, without GI symptoms. Again, this is a small pilot study with 18 children with autism, 20 typical kids, but it's very, very promising. So just to show you what this might look like when we go to 5 metabolites, and then we do some advanced double checking, we then find the red are the metabolites is a plot of the children of the metabolite score for children with autism, and the blue is the metabolite score for the typically developing children. And we see these 2 distributions are quite different from one another. So we just build a mathematical model of those 5 different metabolites together. After treatment, after just the first couple weeks of treatment of vancomycin, we see that the red curve for the kids with autism is now shifted over greatly to the right. So now the metabolites in the blood of children with autism now looks much more like that of typical children. And at week 10, now these 2 groups almost completely overlap. There's very little difference between the metabolites in bloods of kids with autism versus blood of typical children. So we were able to identify that there seemed to be 3 major areas of metabolic disruption involving inosine 5-monophosphate, sarcosine, and tyraminosulfate. Each one of these metabolites correlated with 10 or more others, and they did not correlate with one another. So they seem to really be 3 clues of 3 major different areas of metabolic disruption in kids with autism. And we found that the treatment was able to improve 2 of those 3. So this is a plot of sarcosine. At the start of treatment, the children with autism tended to have very low levels compared to the typical children. After a couple weeks of treatment, it's improved. By the end of treatment, it's improved in all of them. If we look also at inosine 5-monophosphate, at the start of treatment, it's much lower in kids with autism. At the end of treatment, it's much more similar to that of typical kids. So 2 of the 3 improved, but tyraminosulfate, which is right at the limit of our detection ability. Some typical children have higher levels, about half did not. And the children with autism tended to have lower levels. After treatment, there wasn't much change. But it's right at the limit of our detection. So there may have been some improvement that we weren't able to observe. So to give you a little bit more insight, IMP is involved in NAD-dependent oxidation. NAD is a major metabolite in the body, involved in hundreds of reactions in the body. And so IMP is very important for this oxidation type reactions, and also important for xanthosine monophosphate, which is a precursor to making purines. Also, it's been suggested that IMP dehydrogenase activity may be related to irritable bowel syndrome. It's no surprise it's perhaps linked to the GI issues here. IMP is significantly positively correlated with 3-phosphoglycerate or 3-PG and oxidized glutathione. So it's very interesting. So that when it's higher, that tells us that it's certainly related to both energy metabolism, energy generation in mitochondria, and also to folate metabolism. So not too surprising, this connection with both of those very important areas. Sarcosine is important. It's also known as N-methylglycine. It's converted to glycine, which has many, many roles in the body. It's needed to make glutathione, major antioxidant. It's needed to make creatine, purine, serine, nucleic acids, and just general homeostasis of the central nervous system. It's been suggested it may also be beneficial for depression and schizophrenia. So maybe this gives us a clue about a supplement that might be useful. And finally, tyraminosulfate levels. This is the one which did not change much in kids with autism. They were much lower in the group, but 78% of the kids with autism had levels so low that we couldn't even detect it. So you have to be cautious in interpreting this. But we know it's a derivative of tyrosine. It is similar chemical structure to two other tyrosine derivatives, 4-EPS and p-cresyl sulfate. When those two levels are higher, then tyraminosulfate levels are lower. These two chemicals are toxic, that they're associated with much worse ASD symptoms. So it seems that metabolism of tyrosine into tyraminosulfate would be better than metabolizing into 4-EPS or p-cresyl sulfate. Unfortunately, MTT did not improve this within our detection limits. But as the saying goes, 2 out of 3 ain't bad. But it also tells us, gives us important clues of what to look at in future. So just to summarize these results, I'm sorry if they've been a little complicated, but the bottom line is that many metabolites are significantly different in the kids with autism versus the typical kids. We don't know if it's due to the autism or the GI symptoms, but we do know that combinations of just 3 to 5 metabolites allowed us a very high sensitivity and specificity. So we could differentiate the kids with autism and the typical kids with near 100% accuracy. It suggests 3 areas of metabolic disruption, and the MTT improves at least 2 of those 3 areas. And we're still analyzing additional metabolomics results to learn more. But I want to thank the many people who helped us with our phase 1 study and with the analysis of it, the funding from the Arizona Board of Regents for our original study, and some funding from Finch as well for a follow-up study, and for providing the microbiota itself. ARI for funding our early research in gut bacteria. The FDA has been very helpful to us in giving us advice and helping us promptly get our protocols approved by them so we can do the studies. I want to thank, of course, all the families who participated in our study, and thanks Sid Feingold for his pioneering work in microbiota and autism, and Ellen Bolte, who's an autism mom, who's really stimulated the very first study that Sid was involved with. So I also want to mention thanks to the over 1,000 families who have helped us raise over $200,000 for our adult MTT study, and over $1 million for a child MTT study. Unfortunately, due to COVID, our costs have increased somewhat, so additional donations are welcome. And with that, I'll just open it up for questions. So thank you very much.
Thank you so much, Dr. Adams. That was incredibly interesting. Obviously, we've known, kind of like the basics of MTT and the results that you've had, which people have been so excited about. But the metabolites data was incredibly interesting. And do you have any thoughts? Obviously, MTT is kind of at the infancy. Do you have any thoughts on, as we're all waiting, as parents are waiting, you had mentioned that maybe supplementation of some sort might be helpful to increase those levels. Have you guys looked into that, or have any idea on that, that where parents could maybe do the blood test and maybe supplement accordingly?
Yeah, so our blood test isn't commercially available yet, but we hope it will be at some point. We're hard at work on that. I think right now we have a lot to learn. So I would say that people are trying probiotics, and they seem to have some temporary benefit for some individuals. We do have a separate study, I think I mentioned, we're working with Sun Genomics, and that's the largest autism probiotic study to date. Sun Genomics has a special ability to do the sort of testing that we do to evaluate each person's microbiome, and then they attempt to develop a probiotic, a standard probiotic, but tailored for each person's individual gut bacteria. So each batch is made for each individual person. It costs about $350 to be in that study for the initial test, which includes 3 months of the probiotic, and then people are encouraged to retest as well. And if they want to, they can enroll in our study of that. Our study is to simply ask the families to then evaluate their GI and autism symptoms before treatment and again after treatment. And so that is a great way for them to consider. It's primarily available for people in the US, but Sun Genomics is working on extending it to some parts of Europe as well.
That's amazing. Okay, well, I know we have a little bit of time with you. I know you have to leave in a little bit, but let's see if we can get some questions done. This question is from Alex. He had mentioned, you've mentioned that in the past fungal overgrowth is a factor, and you were looking into assessing that. Have you been able to, and kind of what are your thoughts, and maybe what might be helpful as an antifungal, or what are your thoughts on basically the fungal issue with MTT?
Yeah, so I think the fungal aspect is probably a separate issue, but I'll say that based on the paper we published and based on published work by 3 other groups, we can say about 25 to 40%, and I trust the 40% number more, about 40% of kids with autism seem to have a hidden yeast or fungal infection. And our data suggests children with those infections seem to have worse, significantly worse autism symptoms, about 30% worse. And these yeast and fungal infections seem to primarily affect the brain function, but not so much the GI symptoms. So it's very puzzling. It's very hard for people to know that they have a yeast infection just by looking at symptoms, because it just seems to worsen general autism symptoms, especially language and general brain function and cognition. And so the only way to really know is to go ahead and do a test. The problem is the tests have limited reliability. For example, for people who have had such severe fungal infections that they've died from it, even then only about 30% of the tests were able to show a positive yeast infection while they were alive. So detecting these yeast infections can be very, very tricky. But I think every child with autism should be tested for possible yeast infection and treated. The standard treatments are nystatin or diflucan, but certain labs will do a yeast culture and then test to see which antifungal is best for the particular species of yeast in that person. And this is important because some yeast have become resistant to nystatin or some are resistant to diflucan. A study I mentioned, by a survey I mentioned by the Autism Research Institute, suggests that these antifungals are generally very safe and seem to be substantially beneficial. The problem is these fungal infections can often return after treatment stops. So we have a lot more to look into. We have a couple proposals pending to try to do more research in this area, but it just costs money and it's very expensive to analyze yeast. But we are hoping to be able to do a treatment study for yeast as well at some point in the future.
That would be wonderful. Well, we're definitely, I know Enrique is going to put up the information because obviously in order to keep it moving forward, and obviously with COVID threw a monkey wrench in everyone's life, but like same with you guys, we got to keep everything funded, because the more that everything can be funded, we can move forward, we can get on to, results and hopefully approval from the FDA. So hopefully, I'm sure, yes, he put the information up there as well. Let's get to a couple more questions. This question is from Chris. You were talking about, I'm going to mess this up, desulfuribio, excuse me, and its sulfur-reducing bacteria as controversial. Can you elaborate on the possible properties of this bacteria?
Yeah, I think we have a lot to learn about what is the function of desulfuribio in terms of its ability to modify sulfate. We know that sulfate is very unusual in children with autism, that sulfate is a major mineral in the body, fourth most common mineral in the body. Calcium is number 1. Sulfate is the fourth most common mineral in your body. And we know from quite a few studies by our groups and others, about 90% of kids with autism have low sulfate, that they excrete high amounts of sulfate in the urine and have low amounts in their body. Sulfate is important for many functions in the body. Sulfate is important for lubricating the GI tract. And so for kids who have constipation, if you don't lubricate the GI tract with enough sulfate, that could be part of the issue. The good news is it's relatively easy to improve sulfate, either by Epsom salt baths or by giving MSM, which is an over-the-counter supplement. And we demonstrated in our studies of a vitamin mineral supplement that that's a good way to improve it. But desulfovibrio, there's some controversy about whether or not that would be beneficial or harmful. We simply observed that it was lower in children with autism, increased greatly after treatment, and those children, when it increased, along with other changes, was associated with better GI health, better autism symptoms. So because there are other changes going on, we can't absolutely say increasing desulfovibrio is beneficial. We can just say it increased at the same time as symptoms got better. So my guess is it's probably beneficial, but we have a lot more to learn.
Absolutely. I know you've mentioned in the past for Epsom salt baths, do you believe that they have a positive effect on the microbiome, or is that really just more as like the body overall increasing those sulfate levels?
I think it's some of both that I didn't fully explain. In addition to lubricating the GI tract, sulfate is very important as one of a number of routes for detoxification of a wide range of chemicals and toxins our bodies are exposed to or generate. So we need sulfate to detoxify. A simple example is Tylenol or acetaminophen. The standard test for low sulfate is to give that to people and see how long it takes them to detoxify it. And kids with autism take a lot longer to detoxify acetaminophen. It's also needed to deactivate neurotransmitters in the brain. And so bottom line is, yes, I suspect Epsom salt baths would help primarily with constipation. I'm not suggesting drinking it. I'm suggesting bathing in the Epsom salt baths will help indirectly by getting more sulfate into the body. And I think it will also help with, as I mentioned, general detoxification and with improved neurotransmitter levels.
Perfect. And you had, on the graphs, you had mentioned how low some of the kids were, or a lot of the kids were, in Prevotella. And then, but you had, obviously in the graph, you could see there was some Prevotella, et cetera, in some of the kids. Do you remember, because obviously there's more than one Prevotella species, do you remember the ones that the children had that were Prevotella versus the others? And was that able to increase? I think I might have missed that, but was that able to increase post? I think it did. Yes, post. Yes.
So what with this study, we were only able to look at the general. We weren't able to get down to the species level of Prevotella as we were in our previous study. Methodologies changed. So all we can say is that 15 of 18 kids with autism had undetectable levels, and it increased. These 3 who had high levels was kind of puzzling. So I think it's very reasonable to suspect they were less important species of Prevotella, but we don't know for sure.
Totally makes sense. Okay. Here's another question. I think probably have time for 2 more. This one says, I've always wondered what explains the lower number of bacteria species, not just populations, but species in children with autism. Beyond supplementing the missing bacteria through MTT/probiotics, what explains the lack of missing bacterial species to begin with?
Yeah. So we don't know for sure. There are probably multiple factors. One of the factors is just when you have an overgrowth of one particular species, it tends to crowd out the other species. So it tells us that you have an imbalance in your bacteria. An analogy I like to use is, let's say you have a village, and your village has a certain number of teachers and doctors and farmers. And if everyone's a farmer, that's not very good because there's no one to heal you if you're sick. And if everyone's a doctor, that's not very good because there's no one to grow your food. So you need a balance in your village. So our gut bacteria do many, many different things, and we want to have a good balance of them. And that's again why I'm not so keen on regular probiotics, because what I think is better is to try to have a balance, not just a high amount of a few individual species. So that's generally what we're trying to go for. But one factor that I suspect is part of the problem is many children with autism have a self-limited diet and eat only a very narrow range of foods. In general, any dietitian will tell you you're encouraged to eat a wide diversity of foods. We like to say a rainbow of fruits and vegetables. So the more diverse your diet, generally the more types of bacteria that will support. So I think having a more diverse diet probably is one of the factors. But these children in our study didn't change their diets during the study, and yet their bacteria diversity increased quite a bit. And I think that's because we were able to remove some of the overgrowth and just better balance the overall bacteria.
Wonderful. Well, I know you have places to be, people to see. So we're probably going to start wrapping it up soon. I'm just trying to see if there's any more questions that you didn't already cover. Well, this is, I mean, you kind of touched on this, but just lastly, you explained, this person said, when metabolites matched, sorry, this is Ravi, when metabolites matched after treatment, trying to translate this, does that match with the symptoms of autism? So I think what they're asking is, as the metabolites changed, did maybe the autism's symptoms, or did they improve?
It's hard for us to say with this small number of participants. The challenge we have is that 16 to 17 of our children had great improvements in GI symptoms and in autism symptoms. So we don't have many who didn't improve. So we can't, when almost everyone improves, you don't have enough to differentiate what was different about the few who did not. We just don't have large enough numbers. So it's a great question, and we'll try to answer that in our future studies when we have more participants.
Wonderful. Well, we're absolutely looking forward to your next studies. But thank you so much. I know you have to go. Thank you so much for joining us today, and also for the entire audience. Please consider donating to the Brain Foundation, which is a nonprofit organization which helps to just basically fund more research for autism. And also please look into the GoFundMe for Dr. Adams and his colleagues' research, so we can keep funding, keep everything moving forward for our kids, so that they can just have more and more options and more data and more answers. And thank you so much for leading the way on the answers, Dr. Adams. We really appreciate you being with us today.
Thanks very much.
Have a wonderful weekend.